Hello, this is Joe Delaney, creator of SWAN. In this video, we'll be discussing the biological concepts behind SWAN pathway analysis. Canonical pathway analysis treats all genes equally. So in this example of a pathway, there are 5 genes, and in the experimental dataset, 3 genes may be altered in one sample. In canonical pathway analysis, then some sort of background is created. This can be a randomized distribution or some idealistic curve. And then a statistical test is performed to determine if the experiment is different from the random distribution. The interpretation of this is relevant to many biological pathways, but relatively simple. So a reactant transfers all the way to a product, and in this example, the product is transcription. And this goes through a series of mediators. This is a Newton's cradle model. Basically, a reactant bumps into these mediators until it forms a product. Biologically, what does this look like? Well, let's presume the reactant is some sort of cytokine or extracellular signal. It has to get into the cell through mediator A, and then be modified on an organelle, such as mediator B, and then recruited to DNA through mediator C. Eventually, this leads to that product of transcription. This is relevant to plenty of biological pathways, but we do know a little bit more about biology thanks to genomic-wide screens. And from those, we know some of these proteins are actually rate-limiting for a certain pathway. And the way we know this is that if you knock out one copy of these genes, that protein becomes haploinsufficient. So in this example, we can see that it may be very difficult to get transcription if the reactant cannot get into the cell, if it does not go through transporter A. So A is a haploinsufficient gene. How does SWAN incorporate this haploinsufficiency data? Well, that mediator then becomes more important in the pathway analysis. Technically, this is actually a higher magnitude score for everything connected to that gene. This is also incorporated into that background analysis, where your random draws can occasionally be on that haploinsufficient gene, or it can be on normal genes. Aside from those haploinsufficiency data, we also put in regulation data, protein-protein interactions. So in this pathway, we can envision there may be a kinase, which modulates forward flux by phosphorylating many mediators within this pathway. So how does this get built into SWAN? Well, this is why SWAN is a network pathway analysis. We build a network of everything that kinase interacts with and put that into our calculations. And so here the kinase node is connected to 4 other nodes within the pathway, and because of that, its magnitude is much higher than other nodes within the pathway. And so in this example, instead of just having 3 genes that are deleted forming a -3 result, we can actually incorporate those phenotype information to say if the genes that are more relevant for this pathway are altered, then the score is more negative. So instead of a -3, we may get a -6. This isn't precisely how the calculations are done. There's another video on how they are done, but conceptually this is how the math works out. And importantly, with all pathway analysis, the background is really important to eventually determine that p-value. And so we use a random draw process. So this is applicable to any network and pathway. And so as we do our random draws across this entire pathway, we can get different scores, ranging from -1 to -3, with a single gene being deleted on a random analysis. Just repeating that to show you that many times this are on the non-relevant genes, it still creates a score, or the prioritized genes, like the haploinsufficient gene or the kinase. Biology is usually more complex than what we've shown, and so, depending on how much you actually know about your pathway, you are able to incorporate different scores instead of haploinsufficiency. So if you think transporters are really important for your analysis, you can score all those really high, or if you think epigenetics are really important for your analysis, you can score those high within your pathway analysis as well. There are limitations with any pathway analysis software, so they are limited by how the pathways are constructed. And in general, all of these software assume that each gene within the pathway is important for forward flux in the pathway. And so by default, there's not information on positive and negative regulators within a pathway. However, because of this problem, there are pathways that have been created which only mark positive or negative regulators within this pathway. So this is available within SWAN. All you have to do is select the Go Directional pull-down menu. Custom scoring may be applied if you happen to know more about these genes within your pathway, and then you can score all those different genes with positive or negative regulator multipliers. Another limitation is that haploinsufficiency and interactions are often context dependent. And so if you know more about this through screens, shRNA screens, CRISPR screens, about your particular context, you can replace those scoring mechanisms with your own custom lists. Another limitation is that if one gene interacts with everything else within the pathway, this can skew your results to that single gene. And so to remove potential false positives like that, you can actually just remove the interaction data within the analysis. I think one of the frustrating things about this is that to calculate that background, which is so crucial to getting a correct magnitude and p-value, this is computationally slow because you have to do those gene permutations. Of course, we focus on the strengths, and SWAN pathway analysis really shines because it actually incorporates biological complexity into a systematic form of pathway analysis. It's tunable, so like I said, the haploinsufficiency or network building data can be altered by the user, and it's integrative. So if you have more than one layer of data, such as copy number, RNA, and mutation, you can layer those all within the same pathway analysis, or even screen data. The most commonly asked for piece of information by our colleagues who use this software with us is, okay, well, we know a pathway, but what are we actually studying in our experiments? And SWAN is able to prioritize which gene or protein is most relevant to building that magnitude of score within your pathway analysis. What this means is your top hit gene within your top hit pathway can then be knocked down by shRNA or guide RNAs or overexpressed if it's elevated. This is really crucial to following up your pathway analysis in biological experiments. And of course, we think it's really important that this be easily usable by basic scientists. No programming knowledge is necessary. All right, so I hope you've learned a little bit about the biological information which is put into SWAN pathway analysis, and thank you for your time.